Hello, I'm Tim McLaren, and for the past 2 years I've been doing research on the small molecules in our blood in the lab of Jain at UCSD. Um, Today I'd like to share a little of that work with you, and I will couch this work in the larger picture of precision medicine and how emerging technologies are providing um, the tools that we need for this brave new world of medical practice. So this is the way that I see medicine today. We have people who are not well, and we have a variety of treatments that we could give to them. For each person, certain treatments will be more effective than others, and some treatments may actually be completely ineffective, and in fact, some treatments may even be harmful. We're often left to guess which treatment will be best. So we do our best, but we perform ineffective treatments on those who have access to care, while resource limitations leave many others without treatment at all. But what if we knew from the beginning what treatments will be most effective for each person? How would that look? In an ideal world, each person would receive the most effective treatment available as their first treatment. We would not put our patients through unnecessary, ineffective treatments, and we would conserve our healthcare resources so that they could be shared more evenly among the entire population. So this is the hope of precision medicine. And what makes the fundamental difference between so-called precision medicine and the way we've always done things? Well, the goal of precision medicine is to match patients with the treatments that will make them well. But actually, isn't that what we've always tried to do? Um, the difference comes in how we approach the goal. In its ideal form, precision medicine characterizes each patient well enough to understand the causes of disease with molecular specificity. Treatments are targeted to interrupt pathological processes at points where they will be most effective with the least side effects. Again, this is the approach we've tried to take, and we've made some progress, but the problem is that we've largely lacked the tools to characterize patients with molecular specificity. So we do not understand the exact pathogenesis of some of the most important diseases of our time. Read cardiovascular disease, diabetes, cancer, etc. And we do not know why certain subsets of the population respond better to certain treatments. Okay, so let's be a little more precise about what we mean when we say a precise characterization of a person. So let's say this is you. We traditionally take a history, perform a physical exam, and order whatever labs and imaging are necessary. Physicians see, hear, and even smell you as you are, and their understanding of you is generally far more nuanced than they can express in words. They summarize as much of that nuanced understanding in their notes as time will permit, which is usually not much. In your medical record, you end up being represented something like this. But what if we had a more sort of, shall we say, high-resolution way of understanding you and the many facets of you? Um, what do the omics say about us? If a picture is worth 1,000 words, how much is a genome worth? Or what if you knew all 10,000 species of microbes that inhabit your body, or all 4,000 small molecules dissolved in your blood? Emerging technologies allow us to see you from many different angles, so that the version of you stored on the hard drive starts to become much more true to life and much more detailed. What's more, these technologies are inherently digital, so we can put your data into a database with thousands of other people and start to learn what makes the difference between those who get sick and those who don't, between those who will respond to treatment and those who won't. So the first few building blocks of precision medicine are becoming apparent. This will require precise measurement of as many facets of you as possible. It requires participation of other people to begin to see patterns emerge, and that requires one central place to store and access the data. So we can ask, what are some of the most high-yield sources of information that will tell us much about a person's propensity for health and disease, as well as their current state. I've already mentioned a few of them. Let's start with genetic sequencing. A lot of work has gone into understanding the genetic architecture of human disease, and that work is extremely important. And that work is actually accelerating quickly. We saw in 2001 it cost roughly $100 million to sequence a single genome, and prices since then, or the cost, I guess, since then has fallen precipitously to the point that now we're reaching towards $1,000 per genome. And so this is starting to put this kind of like deeply precise characterization of a person into reach of everyday medical practice. So the genome is incredibly important, but actually decades of epidemiological research have shown that the majority of population attributable risk for human disease actually are not purely genetic in etiology. They come from everything else we experience. It's our endogenous physiology, the microbes that inhabit our guts, the food we eat, air we breathe, the liquids we drink, and the drugs we take. All these things together contribute. The problem is it has been notoriously difficult to measure these experiences. But these experiences exert their influence, by and large, by the introduction of small molecules into the bloodstream, which then go on to exert their effects in three main ways. They participate in chemical reactions, cell signaling, and in metabolic balance. And so we can use the small molecules in blood then as more specific measures of our experiences. And we can associate these with outcomes of health and disease to start to learn what matters, and perhaps even why. So the goal then becomes to take a comprehensive view of the small molecules in blood across thousands and thousands of people. And mass spectrometry gives us the most comprehensive view we can get out of any technology available. It can measure thousands of molecules in blood, but the problem is that traditional methods have been too slow and correspondingly expensive to apply on a population scale. This kind of analysis takes on the order of 100 minutes and $1,000 per sample to analyze. So we estimated that in order to do this kind of analysis on a population scale, we would need to improve throughput by 2 orders of magnitude, going from 100 minutes per sample down to about 1 minute per sample. So we've done 2 things differently to address these issues. First, we've automated sample preparation and introduction of samples into the mass spectrometer. And second, we use a very abbreviated form of chromatography, the specialized column, that allows us to separate the chemical complexity of blood across a much smaller time than traditional liquid chromatography would allow. Now, for this to be applicable in a biomedical setting, it has to be extremely robust. We've applied our assay to over 600 different chemical standards, and we see that we're detecting about 70% of them across a wide variety of chemical classes. We can measure linearly across 4 orders of magnitude in concentration, which allows us to make fair comparisons between the levels of a given compound in my blood against the levels of that same compound in your blood. When we measure the same compound multiple times, we get comparable results. And when we measure when we measure the same sample multiple times, we get comparable results. And when we measure this thousands of samples, the results of the first are still comparable with the results of the last. Now, all of these things, though, are just prerequisites. The critical advance is this. We can now do this in under 1 minute per sample, which allows us to process over 1,000 samples in a day. And this is absolutely critical because it allows us now to do this kind of analysis on a population scale for the first time. So now that we can measure thousands of things in thousands of people, we can start to ask questions like, which molecules in blood associate with cardiovascular disease? This is a modified Manhattan plot with the inverse log of the adjusted p-value for a range of masses that have been measured. And so towards the top of the plot are those molecules which associate most strongly with cardiovascular disease. And we saw two molecules that associated particularly strongly with cardiovascular disease. These were found in all study participants. So I assumed, I thought, these are probably endogenous molecules. And what made them even more interesting was that they don't just associate strongly, they associate inversely with the number of cardiovascular events. So the healthiest people actually have the highest levels of these in their blood. And this is really intriguing because if it's a deficiency that leads to cardiovascular disease, that's very treatable. If that's what's happening here, that's very treatable. So we really wanted to know, what are these things? And what might they have to do with cardiovascular disease? So we looked up the masses of these compounds in databases of known compounds, and that returned no results. So we took the molecule and we fragmented it using a mass spectrometer, and then we measured the masses of the resulting fragments. And this is what we observed. We saw that both molecules actually shed some of the exact same fragments. Because molecules fragment in very predictable ways, this means that these two molecules are structurally very similar. And when we looked for the masses of these fragments in a database of known compounds, we saw that these last two were actually the masses of stearic acid and linoleic acid. So we have two structurally very similar molecules. Both have fatty acid chains, and both are associated very strongly and inversely with cardiovascular disease events. So we were really intrigued, and I spent probably a couple of weeks using chemical drawing software to try to piece these things back together, unfortunately to no avail. And this is actually one of the biggest problems in the field. The metabolomics studies regularly find thousands more compounds in blood than they can identify. So to address this issue, a postdoc in the lab had developed a way of where we could take our fragmentation spectrum and blast it against a database of over 1.3 billion spectra from known compounds. And doing this, we found a hit. It matched to lysophosphatidylcholines, or LPCs. So what are lysophosphatidylcholines, and what might they have to do with cardiovascular disease? Well, lysophosphatidylcholines are constituents of membrane phospholipids, which are cleaved by phospholipases to form fatty acids and the lysophosphatidylcholine. And these exist in a dynamic equilibrium with membrane phosphatidylcholines. LPCs have been shown in the literature to bind with about 8 different G protein coupled receptors on a wide variety of cell types. In fact, some authors believe that every cell in the body can respond in one way or another to lysophosphatidylcholines. Given the number of ligands, receptors, and cell types involved, it's perhaps not surprising that the biological activities reported for these molecules are various, and at times even functionally antagonistic. But since we're measuring these in blood, we can ask, what's the net effect across the entire organism? What's the net effect of these molecules? Well, we've seen an association which would be consistent with a protective role for these molecules. This, of course, does not imply that they are causing the protection. But if these molecules were causative for protection against cardiovascular disease, then we might also see that they may protect against some condition or some state which precedes cardiovascular disease. And in fact, we do see a very strong inverse association with 3 different markers of inflammation. This, of course, does not prove anything, but it begins to focus us on a mechanistic hypothesis. Now, it'll be really important in coming studies to look at the timing of the decline in LPCs, the rise in inflammatory markers, and the incidence of cardiovascular disease to see if the temporal profile matches what we would expect for a chain of causation. And that'll be really important to look at that so that we don't end up spending a lot of time on this hypothesis when in fact it could be quickly proven false. But let's step back for a minute. We've measured the small molecules in blood to get a comprehensive view of the internal milieu. We've associated these with outcomes of health and disease, and we've begun to focus and form mechanistic hypotheses. But let's go further back in the chain than that. If the decline in these molecules is not just a result of cardiovascular disease, then perhaps there are certain environmental factors that influence the serum levels of these biologically active forms of these molecules, of these LPCs. What if there are certain microbes in our guts, for example, that produce variant forms of lysophosphatidylcholines, which then go on to be absorbed into the bloodstream and perform some biologically important role? We have both stool and serum samples from the same patients. So we'll measure the small molecules in stool, use spectral alignment to find novel variants of lysophosphatidylcholines, and then we'll sequence the microbiome to see if there are certain microbial communities which associate with specific variants of these molecules, of these lysophosphatidylcholines. Then we'll measure them in serum to see if they make it into the bloodstream, and we'll compare the results with clinical outcomes to see if some microbially produced lysophosphatidylcholines are mediating some of the effects of our health. Okay, so that was we've gone through some of our own work now in metabolomics, or the study of the small molecules in our blood and other biosamples. And we've seen that the cost of genome sequencing is falling precipitously. So we're beginning to build the tools of precision medicine. But here's the issue now. We're generating a massive amount of data. The genome has about 3 billion base pairs in it. The microbiome has an estimated 10,000 species that inhabit a given person's body. And we're measuring 2,000 to 3,000 chemicals in the blood of any individual. And then, of course, in the traditional clinical data, you have natural language descriptions of almost anything. A doctor could describe anything about their patients in the medical records. And then you have imaging data on top of that. So we have a massive amount of data, and it's not just once for each person. We might collect these data sets multiple times across a person's lifetime. So we have so much data, and it's far more than a human brain can comprehend. You can't take in all of that at once. And the way we've typically dealt with that is we've just looked at individual genes, individual microbes, individual chemicals in our blood. This very sort of reductionist approach that we've tried to do. And that has given us some progress. But precision medicine will really come into its own when we can start to look at all of these things together and start to appreciate that a biological system is just that. It's a system, a web, not just a chain, but a web of causes and effects. And so how can we take all of that together as a whole and start to pull some meaning out of it, to translate all this data into meaning? And let's be a little more specific about what meaning we want. For me personally, I want to see a description of the pathology, and I want to see treatment recommendations. I want to know, how does this thing work? How does this disease process work? and where is the most effective place for me to intervene to return a person to health. So this is sort of the output that I want the machine to take all these data that we're feeding it and translate it into an output that looks something like this. That would be my preference, my goal. Now, Google has found a great way to approach translation of very large amounts of data. It's called Google Translate. So this is their translating meaning from one language into another language. So they're able to take the words of one language, extract the essential meaning from that, and then take that meaning and translate it into the words of a different language. And this is perhaps not so different from what we'd like to do here. So let's see how they approach this.
Google Translate is a free tool that enables you to translate sentences, documents, and even whole websites instantly. But how exactly does it work? While it may seem like we have a room full of bilingual elves working for us, in fact all of our translations come from computers. These computers use a process called statistical machine translation, which is just a fancy way to say that our computers generate translations based on patterns found in large amounts of text. But let's take a step back. If you want to teach someone a new language, you might start by teaching them vocabulary words and grammatical rules that explain how to construct sentences. A computer can learn a foreign language the same way, by referring to vocabulary and a set of rules. But languages are complicated, and as any language learner can tell you, there are exceptions to almost any rule. When you try to capture all of these exceptions, and exceptions to the exceptions, in a computer program, the translation quality begins to break down. Google Translate takes a different approach. Instead of trying to teach our computers all the rules of a language, we'll let our computers discover the rules for themselves. They do this by analyzing millions and millions of documents that have already been translated by human translators. These translated texts come from books, organizations like the UN, and websites from all around the world. Our computers scan these texts looking for statistically significant patterns. That is to say, patterns between the translation and the original text that are unlikely to occur by chance. Once the computer finds a pattern, it can use this pattern to translate similar text in the future. When you repeat this process billions of times, you end up with billions of patterns and one very smart computer program. For some languages, however, we have fewer translated documents available, and therefore fewer patterns that our software has detected. This is why our translation quality will vary by language and language pair. We know our translations aren't always perfect, but by constantly providing new translated texts, we can make our computers smarter and our translations better. So next time you translate a sentence or web page with Google Translate, think about those millions of documents and billions of patterns that ultimately led to your translation. And all of it happening in the blink of an eye. Pretty cool, isn't it? Give it a try at translate.google.com.
This is an IBM-built supercomputer named Watson winning Jeopardy against two of the two reigning Jeopardy champions of the world. They fed into the computer question and answer pairs from previous Jeopardy questions. And then they gave it access to basically all of the world's knowledge through the internet. And they allowed the computer to learn how question and answer pairs are structured, how do you sort of extract meaning from the World Wide Web, and then translate that meaning into whatever the answer is that the questioner was seeking. After winning Jeopardy, IBM took Watson and started to apply it in a medical setting.
Sloan Kettering is one of America's first cancer centers, and our mission is to care for people but also as part of the process, always looking to find a better way to do things.
But the massive amount of data that we collect is difficult for any one person to analyze.
So having the information and the wisdom at your fingertips is going to basically make every doctor the most experienced doctor in the world for taking care of that particular problem.
The partnership between Memorial Sloan Kettering and IBM Watson is really at the forefront of our ability to deliver evidence-based medicine through analytic approaches to understanding cancer as a disease.
What our role is that we are MSK Watson's teacher. And just like we would teach a trainee in medical oncology or surgical oncology, we're teaching the MSK Watson system. One of the ways that IBM Watson won Jeopardy was that it was fed in a lot of prior Jeopardy questions and answers, and it learned the correct way to answer those. In essence, MSK Watson does the same thing, but the information we're feeding into it is how Memorial Sloan Kettering doctors treated Memorial Sloan Kettering patients.
Watson's capability to analyze huge volumes of data and reduce it down to critical decision points is absolutely essential to improve on our ability to deliver effective therapies and to actually disseminate that information to the world.
It has access to research. It's taught by specialists. It's also taught by the experience of the patients treated at specialty centers like ours. Watson learns from those cases and gives you a list of choices really chosen for you, which is something we've never had access to before.
I think this is beyond an evolutionary step. I think this is a revolutionary step. This has the potential of totally changing the way we conduct medicine.
We need to get our best to other doctors so that patients everywhere can take benefit from that.
Will allow us to design treatment programs and also ways to follow patients that avoid over-testing and to more effectively use a treatment to personalize their therapy.
Now, I'd like to say that there's two parts of any medical problem. There's the biology and there's everything else. What Watson's going to be able to give us is the everything else part, and that's going to make a total comprehensive package that's going to improve results worldwide.
I think Watson has a bright future in the brave new world of precision medicine. This is one other thing that I found incredibly striking. A few months ago, I saw Craig Venter present at UCSD, and he showed us this really, really cool technology that they're working on, where they've taken 3D images of people's faces, and then they've sequenced the genomes of those people. And then they've used machine learning algorithms, not too different from Google Translate or IBM's Watson. They've used machine learning algorithms to understand how do you go from a genome to the 3D projection of a person's face. And so they've fed in, as input, many faces and many genomes. This face that we're seeing here was not fed into the system. Only the genome of this person was fed into the system. And then the system was allowed to predict what this person's face would look like. And so this is the actual scan of the person's face. This is not something that the system saw. And then based on this person's genome, this is the prediction. Now the fact that we can even get something that looks remotely like a face is really incredible to me. That from a genome, we can ask a machine to learn how genomes work so well that it can actually predict a face structure based on that is just amazing to me as a biologist. But the fact that it looks so similar to this person is even more striking to me, that this is actually, it's not just predicting that, like, yes, okay, you can make a human face, but you can actually make this person's face. It's almost, you could pretty easily recognize this person based on the projection predicted by the genome. It's absolutely striking. So these are some of the tools that I think are beginning to develop behind precision medicine. And so let's add to our building blocks of precision medicine machine learning and computational power, because this takes a lot of computational power to do this kind of analyses, this kind of machine learning. And I'd like to finish just with a vision of the future. Future certainly could go any number of ways, but this is one vision which I find exciting, and I would like to have my medical practice look something like this. So when you go to the clinic, you give samples of your blood, urine, and feces. Automated laboratory systems perform deep sequencing on your genome to determine your genetic background, and on your lymphocytes to determine your current adaptive immune sensitivities. They sequence the microbes in your stool to determine the composition of your microbiome. They measure the thousands of small molecules in your blood to characterize all of your experiences, to see how life has been treating your body. All of these data are placed alongside data from millions of other people in a centralized database, where machine learning algorithms reconstruct the complex hierarchy of patterns that make your body what it is. Back at the bedside, your physician enters your chief complaint into the system, and it responds with a specific explanation of what is happening inside of you, based on your own data. If additional data are required to make a definitive diagnosis, the system knows which questions to ask and which tests to perform. Just like your GPS, the system takes your personal data and plots the most effective course of treatment to return you to optimum health. You and your physician discuss it. If some part of the treatment plan is not acceptable to you, then your physician tells the system to reroute for the next best course. An app on your phone provides reminders to help you follow your treatment plan, and it checks in to see how well you're doing. Your physician keeps an eye on your responses and follows up as needed. This is how I see the clinical side of precision medicine going forward. Now, meanwhile, scientists analyze the hierarchy of patterns in the database to understand better what drives health and disease. Those patterns which demonstrate the strongest correlations with important disease states are prioritized, focusing our collective efforts where they can lift the greatest burdens from society. The same algorithms that plot out a patient's treatment plan also predict new interventions. Scientists develop ways to deliver these interventions, and physicians implement them. With each visit of every patient, data are added to the system, and our collective intelligence grows. This, of course, is just one vision of the future, but it's a vision which I am excited to continue to build going forward. I have a lot of thanks to give for these 2 years that I've been blessed to spend doing research. The Sarnoff Cardiovascular Research Foundation funded me for this research and helped me to find my way to the lab of Mo Jain. This all started with Amy Davidoff, who was my sponsor at my medical school, the University of New England. I've been doing my research at the University of California. This is Mo Jain's lab here on the top picture, and that's Mo in the center. He's a brilliant scientist and a good friend, and he's shown me how to focus my curious energy to productive ends. On the far right of this picture is Jeremy Watrous in the plaid shirt, and he's also a brilliant scientist, an excellent mass spectrometrist, has taught me the great majority of what I know about mass spectrometry, and he's contributed significantly to the mass spec projects that you saw in this slideshow. The rest of the members of the lab have been absolutely fantastic, a great support, and it's fun to work with them too. Anyway, thank you for your time and attention, and I hope to work with you to build to build precision medicine. Thanks.